Okay, we've talked about enzyme catalyzed reactions and how they can be described with the Michaelis-Menten equation, and we've talked about different kinetic constants that can be used to help quantify that enzyme activity. Things like Km, Vmax, turnover number, catalytic efficiency. Now we're going to move into a talk about inhibition and drug inhibitors. And we're going to look to see how different drug inhibitors affect these kinetic constants. And the reason why we think this is important is because a lot of drug inhibitors are, they're drugs, they're pharmaceuticals. And the inhibition of enzyme catalyzed reactions can sometimes dampen biological processes that are causing or contributing to disease. And so there are a lot of pharmaceuticals that act as enzyme inhibitors. This slide has a lot of information on it, but it's a good one-page look at the different types of inhibition that we're going to look at. Irreversible inhibition, where the inhibitor binds to the enzyme irreversibly with a covalent bond and does not detach. There's a subset of irreversible inhibition called suicide inhibitors, and we'll talk about those. And then the rest of the inhibitors are reversible inhibitors. There's the competitive inhibition, uncompetitive, and non-competitive or mixed inhibition, and we'll talk about each of those. The most common type of inhibition in pharmaceuticals are probably irreversible excuse me, and competitive inhibition. So let's start by looking at an example of irreversible inhibition, and it's one you probably are familiar with. Let's look at this animation here that illustrates the action of aspirin on the enzyme cyclooxygenase 2, or COX-2. And so aspirin is a member of a family of drugs called COX inhibitors that prevent inflammation through inhibition of this enzyme. And it does lots of other things too. So here's an example of irreversible inhibition. Here is the COX-2 enzyme in blue, and here's an important serine in the active site. And what we are going to see is that aspirin, right, is going to actually react with the serine in the active site of COX-2 and form a covalent bond with it. Now this inhibitor is going to form this covalent bond, and that is an irreversible reaction. It doesn't just bind due to electrostatic or hydrogen bonding and then dissociate and bind and dissociate. It reacts with it, and it really irreversibly clogs up the active site of this enzyme. So let's look at that happening. Right? The alcohol group on the serine is going to attack the carbonyl group of aspirin and form a covalent bond. Boom, there it is. Right? The rest of the aspirin molecule dissociates, but it has irreversibly modified the active site of the enzyme, and that's why we call it irreversible inhibition. Now, we said that there's another type of irreversible inhibition called suicide inhibition. That's when these inhibitors bind covalently to the active site, and they're modified, but the entire inhibitor stays covalently bound to the active site. They sort of dive in like a kamikaze in a jet plane, react with that active site, and stay in there, right? Suicide inhibition. And again, this is a type of irreversible inhibition. Now let's look at this animation and talk about some different types of reversible inhibition. Let me skip through some of this. Here we go. Here are the three types of reversible inhibition. Now this is a lot to look at too, all of these things floating around and moving. But the first thing to notice is why this is reversible as opposed to our irreversible inhibition. In all three of these cases, competitive, non-competitive, and uncompetitive, you can see the inhibitor drug, and it binds and it dissociates. It binds and it dissociates. It does not form a covalent bond and stay irreversibly stuck to the enzyme. There's some equilibrium constant, some big K value, that describes the binding and dissociation of these inhibitors. And so let's go through them one by one and see what the difference is. Competitive inhibition is the most common type of inhibition, and it's the easiest to conceptualize, really. If we see here, there's this red inhibitor, and it looks a lot like the substrate. And in fact, when the inhibitor interacts with the enzyme, it binds to the same place that the substrate's binding to. And when the inhibitor is bound, the substrate can't bind. And so the substrate and the inhibitor are competing for the same binding site on the enzyme. And obviously, when the inhibitor is bound, the substrate can't, and the enzyme isn't working. So that's the mode of inhibition for these competitive inhibitors, is they clog up the binding site for the substrate and compete for it with the substrate. So good competitive inhibitors are ones that bind really well to the enzyme, have an equilibrium constant where it's often bound to the enzyme to form an enzyme inhibitor complex. And, it would be nice if you had a high concentration of this inhibitor, because then it would be able to outcompete that substrate. So competitive inhibition is pretty straightforward. It's a competition. Non-competitive inhibition looks different immediately. Here it is. Here's this inhibitor that's shown to be binding to a location on the enzyme that's different than the substrate binding site. It's binding in a different location, and when it binds, it's causing interference, right? When the inhibitor is bound to the enzyme, the reaction doesn't proceed towards product formation. Now, in non-competitive inhibition, this inhibitor can bind to the inhibitor binding site of the enzyme whether the substrate is bound or not, right? So it doesn't care whether the substrate's bound. Now, in uncompetitive inhibition, we have a similar situation where the inhibitor is binding to an off-site location, but in uncompetitive inhibition, it can only bind when you have the enzyme-substrate complex. When the enzyme is free enzyme, that binding site is not available to the inhibitor. So you can think of this uncompetitive inhibitor as it really kind of locks up the enzyme-substrate complex, making it unproductive. Now, here's a table that we're going to revisit after we go through some more information about these types of reversible inhibitors that really describe how each of these inhibitors can affect, Km and Vmax. And this is really important, because if you want to find out how your drug is interacting with the enzyme, you can do some experimentation and look at how this enzyme catalyzed reaction proceeds in the absence and presence of your inhibitor, see how it influences Km or Vmax, and then you can tell how it's working, what its mode of action is: competitive, non-competitive, uncompetitive. Okay, so let's look at some of the details of competitive inhibition. As you'll notice here, let me get a pen. Here's our Michaelis-Menten equation up here, which should be familiar to you. This is the basis for our derivation of the Michaelis-Menten equation. Enzyme plus substrate goes to the complex and proceeds towards product. And here's how the competitive inhibitor interacts. It binds to the free enzyme, right? And really this free enzyme has a choice. Is it going to bind to the substrate? Is it going to bind to the inhibitor? And that's going to depend on its affinity for each one of these and the concentration of each one of these. So when the enzyme binds to the inhibitor, it forms an enzyme inhibitor complex, and that's not a productive complex. Now we can describe sort of the affinity of the enzyme for the inhibitor with what we call a Ki value, or dissociation constant, which is an equilibrium constant, which describes this dissociation reaction of the enzyme inhibitor complex. So it's the free enzyme and inhibitor as products, the enzyme inhibitor complex as the reactants, and obviously if you have a high dissociation constant, really it's showing a weaker interaction between the enzyme and the inhibitor, spending most of its time separate. If you have a really low dissociation constant, that indicates a really potent inhibitor. A low dissociation constant means that the enzyme is binding to the inhibitor most of the time. And so an effective inhibitor is one that has a low dissociation constant, and you've got it at a high concentration, and it can outcompete that substrate. Now, what happens to our enzyme catalyzed reaction in the presence of a competitive inhibitor? Well, this is the way the curves sort of change in our Michaelis-Menten plot. Here, this sort of greeny-yellow line here is a plot in the absence of any inhibitor. This is just the enzyme and the substrate making product. And then the blue and the pink here are with increasing amounts of inhibitor. Now, when we add inhibitor, it alters the shape of this curve and alters where it is on the graph. And the more inhibitor we have, the more it alters it. Okay? So we're going to start thinking of something called an alpha factor. The alpha factor really describes how much your normal curve is altered or deviated from the norm. The higher the alpha value, the more deviation there is from the normal curve. And the alpha factor depends upon two things, right? One is how much of that inhibitor is in there. The higher the concentration of the inhibitor, the more you're going to screw up your reaction. That makes a lot of sense. The other thing that influences alpha is the dissociation constant. Really, the lower the dissociation constant, the better your enzyme binds to that inhibitor, and the more it's going to screw up your reaction. So we can see that our curve here is altered, but it's going to be hard to calculate something like alpha, just like it was hard to calculate things like Vmax and Km. And so we're going to have to turn this data into a Lineweaver-Burke plot. So if we look at a Lineweaver-Burke plot for a competitive inhibitor, what we see is an interesting pattern here. Here, again, in the green is our normal, non-inhibited enzyme reaction. And when we add inhibitor at low or high concentrations, that line deviates. And what's deviating is the slope. What you'll notice here is that all three of these curves share the same Vmax, or 1 over Vmax. It's just their slope's different. Now what that means is that for a competitive inhibitor, that Km is changing. Vmax is not changing. Okay? And the reason for that is if we look back at this curve, we can say that all of these curves are approaching the same Vmax, and we can always outcompete our inhibitor. If there's a certain concentration of inhibitor in there, we can still reach our maximal velocity by just dumping in more and more and more and more substrate until, for all intents and purposes, our enzyme only sees the substrate. We've outcompeted the inhibitor. Now, it takes a whole lot more substrate in order to reach that Vmax, and that's why Km increases, and we see differences in slope for a competitive inhibitor. Okay. A lot of pharmaceuticals, again, are competitive inhibitors, and if you work for a drug design company, it's sometimes the most obvious thing to try, right? If you know what the substrate is for a particular enzyme and you know the shape of that substrate, then it's kind of straightforward to try to design molecules that have the same shape, the same chemistry as the substrate that would be able to compete for that enzyme binding site.